So good afternoon. So the task that I've been given is to answer all of the remaining common challenges with CAR T cells in 15 minutes, which of course cannot be done. So the one aspect that we decided to focus on is patient access and and manufacturing of CAR T cells to accommodate, but of course we'll also be open to additional questions if you have them and answer them if we can. So the disclosures are here. So of course this aspect of manufacturing is critical. There's a strong desire to scale up manufacturing so that more manufacturing slots are available, to also expedite manufacturing so that the overall wait time for patients can be reduced. I will show though that there's also additional factors also related to the topic of patient access, and that is related to patient education, also healthcare professional awareness that CAR T cell therapy exists, where referral centers are all available to recommend patients for this treatment if at that particular site or maybe in an outpatient setting this is not accessible. And of course, reimbursement is a strong topic. So the certainty and confidence that treatment will be reimbursed, and I think is a critical factor and determines whether patients, in fact, do get access to CAR T cells or not. I'll show you towards the end of the talk, there are also some kind of technical highlights that we currently have in mind in order to really scale up. But let me start, but just reminding, of course, everyone that what you are aware of, that there are a number of approved CAR T cell therapies now, that there is a steadily increasing number of CAR T cell centers across Europe, that there's still a steadily increasing desire from patients to benefit from these treatments, and that even though there's clear recommendations, there are approvals, it's still the fact that only about a third of the patients that, as per label, could get the treatment do receive the CAR T cell infusion. There's, in addition to the scientific and medical factors, of course, these formal and structural factors, let me call them that, and this is clarity on reimbursement and also a commitment from the healthcare system in different EU member states and healthcare insurances to actually cover this treatment. There is some bureaucratic burdens that need to be addressed, and overall I think it's fair to state that there is some inequality also across Europe between different European countries in what patient access to this treatment is. Looking at clinical trials, and you've seen these clinical trial roadmaps in different contexts, it's clear that there's the strongest availability of these trials in Asia and in North America. In Europe, this has improved. However, when you look and compare this to the type of studies that are available compared to the United States, you see that there is fewer phase 1 trials. And it's because we know that in phase 1 it's often about dose escalation and determining a recommended phase 2 dose. Still, because these are living drugs, also a relatively low dose can be to the benefit of patients. So I think we should still aspire and continue our effort in Europe to make sure that we remain attractive as a site for clinical trials, be that from major pharma and from biotech, but also that we continue our efforts and bring the many innovations that are available at the academic sites into first-in-human trials in the IIT setting. That's something that we have, of course, in our heart, and that our site in Würzburg is pursuing along with many colleagues in the field. So I want to highlight the study that was conducted by the Italian group. This again illustrates the challenges of patient access on the example of large B-cell lymphoma. So this is focusing on the year 2020. The study was published in 2021. So some of the challenges that were described here may have been addressed already or may be improving, but I think in principle still it illustrates what challenges we collectively as a field should continue to work on. So it was estimated that in that year in Italy, about 4,000 new cases of large B-cell lymphoma would occur, and that approximately 600 patients should be eligible for treatment with CD19 CAR T cell therapy as per EMA label of the approved CAR T products. There are some criteria that IFA, the Italian regulatory authority, is applying, such that approximately 300 patients would then remain that should de facto be eligible, and also reimbursement would be reassured by the Italian healthcare system. It was then, however, also determined that only 110 patients received the CAR T-cell infusion. So about 2/3 of the patients still got lost in the process. And the authors describe the challenges that they accounted for this loss, mainly to patient and healthcare provider awareness that this treatment does exist and is accessible. Challenges with regards to referral if patients were not close by or lived in a city where a major CAR T center was located. And again, issues related to reimbursement. So the time required to ascertain reimbursement from the healthcare insurance was also indicated as a major barrier. So some of these challenges may have been improved, and also 2020 was the first year with major impediments due to the COVID pandemic. But again, it illustrates that, of course, patient access is not just about improvements in manufacturing, but also related to many structural changes, and beginning at education and information that is still indicated to make patients and healthcare professionals alike aware that these treatments exist, that what their current indications are, and what the reimbursement situation will be. A strong aspect that I think is on people's mind is the question, where are CAR T cells manufactured? Right now, manufacturing is done in these gigafactories where leukaphereses are obtained at the site of treatment, or then cryopreserved or shipped fresh to the centralized manufacturing site once a manufacturing slot has been procured. The manufacturing then takes place. The CAR T cells are then cryopreserved and wait for release testing, which typically, in addition to the typical 2-week manufacturing time, adds another 2 weeks to wait for sterility testing and certain other markers related mainly to vector copy number and genomic insertion. And then the cell product is being transported back to the treatment site. And this, of course, is a lengthy time period that then elapses. It's many weeks, typically anywhere between 1 and 3 months that can elapse, and this for many patients is too long to wait, and this certainly contributes to our losing of 2/3 of the patients that are in principle eligible to receive CAR T therapy, but then not getting it. So one potential solution is to foster point-of-care manufacturing. So this is enabling certified cancer centers to manufacture the CAR T cells on site. This will ease the logistics. This will also ease the cost, and it can certainly contribute to making CAR T cell therapy more accessible. It requires, however, that the manufacturing process is such that it can be conducted in the GMP cleanroom facilities that are available at many university hospitals. That would likely be made easier if there is a virus-free gene transfer, because the handling of viral vectors in GMP facilities then limits their use for other ATMPs. So there's certainly a lot that will be a critical aspect to ease the manufacturing, and also implement automated manufacturing devices, as I will show you, to make this compatible with the staff and the expertise that is available at these sites. So I will show you 3 examples that we think can make an impact on improving manufacturing and thereby contribute to increased patient access in the future. Let me start very high level with a big topic right now that is discussed heavily in the European Union. This is artificial intelligence. It's a big debate now. How do we use AI? Is it acceptable to use AI, and what's our relationship with this? So we are currently engaged in a European project. It's called 8Path, and the ambition is to create the smart hospital of the future. And this is, at the core of this, is an automated manufacturing platform to make CAR T cells, where we're now looking for aspects of how artificial intelligence can help us to manufacture the cell products consistently in best possible quality, and also consistent with the resources that are available at academic sites. So I will show you the use cases that we've identified, and you see from this that, of course, AI is not there, but we're working towards AI, and you also realize that, of course, in order to implement AI, you will need a lot of data, very well curated data, and this we're currently also developing concepts to determine how can we obtain these data in order to make the AI meaningful and impactful. So one aspect is that in the manufacturing platform, and using this manufacturing platform, we're implementing a so-called digital twin in order to allow us to learn along the manufacturing from which patients is the manufacturing going well and from which patient is it not, and how can we kind of pre-identify before we even start the manufacturing, what the yield of the CAR T cells will be, and whether we can potentially still obtain an in-spec product if we adapt the manufacturing. So adaptive manufacturing is certainly also an important aspect. So this leads us also to active bioreactor control. So we're doing online measurements and quality parameters from the cell culture medium, but also from the cells themselves to determine when have they reached the optimal fitness state and are ready for infusion. Not necessarily on a fixed date, as it is currently done, but also acknowledging that lymphocytes from different patients have different intrinsic attributes, and some T cells may be ready after 8 days, other may be ready after 12 days, and of course we would still aspire to reduce manufacturing to as few as possible days, to working towards the enigmatic 24-hour CAR T cell product in order to obtain as much fitness as possible. There's, of course, a big question whether AI can help us in clinical decision-making, and I think this is probably the most challenging because a lot of data will be needed in order to then determine and annotate the clinical outcome, even though there is, of course, a lot of data already there, but clinical outcome data very well curated from many 10,000s of patients will be needed in order to make this meaningful and applicable. And there's aspects of production scheduling and managing resources available at hospitals, including staff, but also the GMP facility itself are included in this. So I think AI has the potential to make an impact, but I think it's early days, and the current work should be about determining where can we implement AI, how can this be done, and is also, of course, asking ourselves, is this ethically acceptable and in line with the high standards of data protection, for example, that we have in place in Europe. A second technology that is real already is virus-free gene transfer. Procuring lentiviral and gamma-rich vector in clinical grade is one of the greatest bottlenecks for scalable manufacturing at this time. So there's a strong interest in going virus-free. The example and the technology that we're using is transposition. So this can be done from mRNA and DNA vectors with the idea that a transposase, this is a cut and paste enzyme shown in orange, is liberating a transposon. This is the stretch of double-stranded DNA shown in green here, which encodes the CAR and is inserting this stably into the T cell genome. We've worked on this technology quite a bit, and if you just look here at the right, I'll show you that we can reach stable gene transfer in a high level of CAR T cells, and we've also put this to the test in a clinical trial. This is the Caramba clinical trial that we're currently conducting in an EU-funded project under the leadership of our institution. Just to show you why this is important, this is looking, of course, also at cost, and we now know that you can manufacture mRNA and DNA at the scale such you can vaccinate half of the planet's population. So the big advantage is here the scale, so that you can, if you have a batch of this vector, you can then trim the cost to less to $1,000 per CAR T cell product that you then make from this, and this compares favorable to the cost of virus that you have to make. So then you gain easily 1, 2, potentially 3 lots of CAR products that you can make from the vector batch that you have available, and that of course allows you then also to multiplex manufacturing in the GMP clean rooms that you have available, and I think this will be overall a big leverage to make CAR T cell therapy more scalable and more accessible from that technological point of view. And one last aspect, I just want to mention it, and there's of course excellent concepts already, and we will see how readily they can be implemented now in the clinical setting. This is the so-called in vivo gene transfer. So this is the creation of CAR T cells without the need for ex vivo manipulation. So the ability to package gene transfer vectors into nanoparticles that ideally are, and if you administer nanoparticles intravenously, most of them will end up in the liver, and this is being used in gene therapy to correct certain hereditary diseases. If you pseudotype them, just as you do with virus, for example, to target CD5, you can also use CD3, CD4, CD8, and in order to then obtain preferential uptake by T cells, you can also then include the genetic cargo you use to encode the CAR in here, and thereby create the CAR T cells in the patient. This is very attractive. There's questions, of course, related to biodistribution, also to safety that have to be answered, but I think there will be certainly be applications where we're going to see in vivo gene transfer for CAR T cells in the future. And then it will be, a simple injection that Uli can administer in the outpatient setting, giving the nanoparticles as an IV or maybe as a sub-Q injection, and the CAR T cells will form themselves in the patient. So I will finish by just reminding you that the T2 Evolve project in the IMI initiative, where Uli is involved, for example, I have the privilege to lead this consortium as the academic investigator, is working on several of these aspects relating to patient information and education. Here are the partners. It's a public-private partnership project with many stakeholders involved. And I want to highlight and encourage you to participate in this survey that we've currently launched. This is trying to capture the patient experience. Patients are invited from every part of Europe to participate and tell us about their experience with CAR T-cell therapy. And this, again, will help us to then improve information material, both for patients but also healthcare professionals. And I think, as we said in the introduction, this shall be an important aspect to also contribute to increasing patient access. So here's the code that you can download from our website. And I do encourage you to motivate your patients to participate and tell us about their story. Thank you.
Thank you very much. You brought us to the next level.
It's there. We just need to climb up.
But there are also already questions. One question is regarding allocars. You didn't allude to those. So how would they fit into the landscape?
Yeah. Allocars is an attractive concept. There's an advantage is that potentially T cells have a better fitness state because these T cells likely have not undergone prior chemotherapy. There are some challenges, though, to this expected outcome of longevity, memory formation that we would hope for from a CAR T cell. Of course, if it's allo, eventually there will be immune rejection, right? No matter if you gene edit the HLA molecules out of there. So I think you're going to diminish the therapeutic window, which can be acceptable in many types of cancer. Also for CAR T applications in non-malignant diseases, the allo approach is very attractive. But we have to be clear that there will eventually be immune rejection, so there will be a different pharmacokinetic with these cell products. And I want to mention that, of course, the different strategies currently being pursued, either taking healthy donors, and then the big question is, what are the best healthy donors? Is everyone equal, or are there super donors? And if so, what are the biomarkers? All these cord blood-derived or iPSC-derived allogeneic CAR T cells. This is attractive, but you also have to keep in mind that if you take an immune cell, go back to induce pluripotent stem cell, and then do the differentiation again to NK cell or T cell and do the engineering for the CAR, these are very long culture protocols, and there are companies that have perfected this, according to the current state of the art, but it's not, don't think they're at a point where they actually improving scale. They demonstrate that this is technically feasible, and the next task will be to really demonstrate that you can now use this to scale up and increase access. That will be the next task. And then the final question is, will they be equally effective? Because if you tell your patient, I have this approved relatively conventional autologous cell product, and that's the performance, and have this allo, which is conceptually cool but a little less effective, which one are you going to choose? Right?
Well, thank you very much, Michael. We have a few minutes for, there were some questions coming in, which we have not answered. So there's one for Dr. Kumar, I guess, and for Dr. Hansen, regarding the BCMA surface expression or protein expression. So does that impact on CAR T-cell outcome?
Yeah, the BCMA expression is pretty uniformly high in all the plasma cells, and there doesn't seem to be any data to suggest that the expression pattern or the intensity on the flow cytometry analysis actually impacts outcomes. On the other hand, there have been several reports of mutations that lead to loss of BCMA expression completely, which has been suggested as a mechanism of resistance in patients who are getting CAR T and then subsequently relapse.
Mm-hmm. Then there are two questions for the lymphoma colleagues. One is regarding IgE replacement. Maybe you could answer. So how do you handle that?
In patients post-CAR T cells.
Yes.
So, I think that we always measure immunoglobulin levels. It's going to be different, obviously, depending on how many lines of treatment somebody has had, how much rituximab they've had before. So I think the only way that you can really handle it is to check immunoglobulins and to, of course, give them if they're clinically indicated and they have low levels of IgG.
And so do you wait until the patient has the first infection, or do you give them prophylactically once, let's say, they drop under a certain level of...
I would check it at every visit, so I would check the levels every 3 months, and if they're, if the IgG is less than 400 and someone is having X number of infections according to the guidelines, then I think they should get replacement IVIG.
The other question was regarding CNS prophylaxis in patients with cerebral involvement. Do we always use Keppra prophylaxis? I would say yes. Yes, we do. I think that's a clear indication, and we should not stay away from it and use it all the time. Well. The good thing is we now have a treatment that really works for these patients with acceptable toxicity and favorable outcomes. So, so we do everything to keep them away from toxicity. I don't think we have any, well, we have several questions, but I think we've answered most of them. There's one more for Michael. The transposon technology is impressive, but is the $1 per dose just the manufacturing cost?
Well, it didn't say $1 per dose.
Yes.
And I don't think that we can do it for $1, but it's less than $1,000 for the gene transfer vector per dose. And I think that's pretty good already. Right?
Yeah, but that's just the production cost or other costs included?
This is to have the vector in hand, and then of course you need to include this into the it's not the cost for to get the car product, it's to get the vector, right? So it's an important factor when you put together the cost to make a cell product, but that's just also, of course, cost related to using the GMP facility, train the staff, which you can also ease when you use virus-free, because you can then shorten the time, you can do multiple products in the same clean room concurrently, so I think this can all go in. I'm not sure if we can make a batch of CAR T cells for less than 1,000. If ever we could, that would be a big advance.